My name is Hannes Svardal. I'm from Austria originally, and I just started in the Department of Biology in October. And I live in The Hague, have 2 kids. Otherwise, I really like traveling and especially enjoying nature in different countries. I really like nature photography, and not only in my work but also privately. I'm a real fish enthusiast, so I like to breed fish at home, but also travel somewhere to find different species of fish. And generally, when there is a river nearby, I really often jump in and just snorkel a bit to see which fish are there. So I started with physics actually at the Technical University of Vienna, and did my undergraduate and masters in physics, and also 2 years in Paris. From there I went on to do theoretical population genetics in the math department of the University of Vienna as my PhD. Then I switched over to analyzing genome sequencing data at the Gregor Mendel Institute, and from there I moved on to study the genome sequences of cichlid fish in Cambridge. This is the Mesodrome. It is a research facility that was built jointly by 2 groups from the biology department. So basically it's a big greenhouse, and the idea is that we have these ponds over here and also raceways where one can basically simulate aquatic ecosystems. The core of my work is actually not experimental, but my background is really in evolutionary genomics. So the idea is you get genome sequencing from many different individuals, and then you look at the differences between them. And these differences tell you something about how they evolved, how they adapted, and how different environmental factors influence their evolution. Cichlid fishes are fascinating, in particular Lake Malawi cichlid fish, on which I work, which have formed more than 600 different species in an evolutionary very short time. Looking at the genomes basically told us that these species, while being quite well-defined species, they still hybridize a lot and they still exchange genetic material. And the system that I want to study is adaptation to heavy fishing pressure. Because Lake Malawi cichlids, they are not only interesting for us as researchers, they are also a very important resource for local people, where fishing accounts to up to 70% of the protein supply of the population. And in some populations, these fish are really heavily exploited. And we see very strong responses in the phenotypes and the characteristics of these fish species. If you look here at the screen, what is scrolling down here is the genomic information of these cichlid fishes. They are actually not analyzed on my computer, but I'm connected here to the VSC, the Flemish supercomputer cluster. So basically I use several tens or hundreds of whole computers that are connected together and that do all the heavy computational work. The whole genome has 1 billion base pairs. In this case, we only show the differences between these 41 individuals. But in order to really understand what is the trait, what is the phenotypic characteristic of the individual that changed, is it that they mature faster, is it that they maybe evolved a way to avoid nets in the heavily fished population? In order to find this out, we need the experiments. And that's why it is so powerful to combine the experiments with the data.